This podcast is sponsored by Salix Pharmaceuticals. Maintaining a healthy gut is key for overall physical and mental well-being. Whether you're a health-conscious advocate, an individual navigating the complexities of living with GI issues, or a healthcare provider, you are in the right place. The Gut Health Podcast will empower you with a fascinating scientific connection between your brain, food, and the gut. Come join us. We welcome you.
Hello, friends, and welcome to the Gut Health Podcast, where we talk about all things related to your gut and well-being. We are your hosts. I'm Dr. Megan Reel, a GI psychologist.
Hello, and I'm Kate Scarlatta, a GI dietitian. Today on the Gut Health Podcast, we're joined by a true pioneer in digestive science, Dr. Mark Pimentel. Renowned across the globe for his groundbreaking work in gastroenterology, Dr. Pimentel serves as the executive director of the Medically Associated Science and Technology, also known as MAST, program at Cedars-Sinai Medical Center in Los Angeles. At the helm of the MAST program, Dr. Pimentel leads an innovative team that's unraveling the complexities of the human gut. Their focus, conditions like irritable bowel syndrome, IBS, a disorder that affects nearly 1 in 10 people worldwide, as well as the lesser known but increasingly important small intestinal bacterial overgrowth, or SIBO, and intestinal methanogen overgrowth, or IMO. Today's topic is the gut microbiome and dysbiosis. Welcome back, Dr. Mark Pimentel.
It's great to be back with you. It's exciting. It's the summertime, but people are still interested in the microbiome.
Who would have thought?
Well, Dr. Pimentel, we want to kick things off today with you with a mythbuster. And, with summer, whatever it may be, what do you think our listeners need to know in the myth-busting world of the microbiome?
I think the biggest myth that we deal with on a daily basis is that the magic is really in the dual microbiome only. And if we're going to talk about microbiome, a lot of what I'm going to talk about is the small intestinal microbiome, which is harder to get to. They used to think that the small intestinal microbiome was sterile. There was no bacteria there, or hardly any. And now we know it's abundant, and the bacteria there are giving you things that you don't want, sometimes giving you things you do need. And that's the myth. The myth is that the bugs that we thought were only in the colon are in the small bowel as well, and they can have create problems for your health.
I often think, are we going to start looking a little closer at the mouth microbiome, the esophagus microbiome, the stomach microbiome, all these places that seem to be inconsequential, but maybe are playing a role, even if the bacteria numbers are vastly different and less, et cetera. But let's kick it off with, what is gut microbial dysbiosis and why should we care?
Dysbiosis used to be a dirty word, or a word that people didn't like to use because it had really no meaning. It just means there's a problem with the microbiome. And that's like saying there's a problem with your arm. That's a diagnosis. No, that's not a diagnosis. There's a problem with your arm. We need something more specific. So I think the term dysbiosis has been thrown around and, again, still doesn't have any meaning. But I think what we're trying to do, or what people are trying to say, is that it's an overarching term that says there's something wrong with your microbiome, and it could be this, it could be this, it could be this. And in each particular instance, it can have different ramifications on your health and your condition. And I think that's really what the term dysbiosis ought to be used for at this point in time. Because, for example, there are things that are advertised. Oh, help your gut dysbiosis. Mm-hmm. What does that mean? I mean, you're selling a product to help dysbiosis, but what dysbiosis are you talking about? So we don't want to use it in that context, but as an overarching term, I think it's fair to say.
Okay. So it's certainly not specific to it's too general for the gut.
Right. Exactly.
Okay, so when it comes to a diagnosis of something that you are studying every day of your life, SIBO, small intestinal bacterial overgrowth, do you believe that this fits the definition of small bowel dysbiosis?
So we're doing a study called the REIMAGINE study, and you may have heard about it, but basically it's the first and largest now study of the small intestinal microbiome, taking aspirates using proper double lumen catheter techniques so that it's sterile. We've been doing this for a while. We have well over 1,000 patients, on our way to 10,000 patients. When you look at the small intestine in a normal person, it barely goes above 10 to the 2, 10 to the 3, or 100 to 1,000 microbes per ml. In the case of overgrowth, it's over 10 to the 3. But that's not the story. The story is in the REIMAGINE data, this is like over 1,000 patients. The most apocalyptic thing we see is overgrowth. And what I mean by that is nothing is as dramatic, nothing is as obvious as when overgrowth is there, because when the E. coli and Klebsiella part of overgrowth blooms or grows, it destroys everything in its path. It's like a bulldozer taking out all the rest of the microbiome. The higher the E. coli is, the more destroyed the rest of the natural garden is destroyed. And so it really is apocalyptic. The only other example of apocalypse on the small bowel is antibiotics. If you take broad spectrum like ciprofloxacin or something like that, recently the microbiome is wiped out. So those two instances are the most obvious dysbioses, if you want to go back to that term. SIBO is very dramatic, very obvious when it's there.
Yeah. So you're basically saying, I just want to kind of recap this for our listeners, that there are certain microbes that you're finding are always there with overgrowth, and they're wiping out some of the other populations. So they're the primary microbes in that condition.
Right. Exactly.
So are they always the bad guys? Like some people have Klebsiella and E. coli and they're fine. It's just when they go rogue in the small intestine.
Yeah, so it's a little bit tricky. So one of the other myths, if we want to go back to myths, going backwards in this podcast, is that even in review articles on SIBO, I used to say, as the textbooks used to say, that it's coliform microbes moving into the small intestine,
Right.
That's overgrowth. Some people even still talk that way. That's absolutely not what it is. We didn't know that then when we were writing these articles. What we now know, it's really, there is a slowing of the small intestine, and that preferentially favors aggressive bacteria, and E. coli and Klebsiella are the two most aggressive. There are others, Aeromonas and other organisms that are minor parts of SIBO, but the Ferraris are E. coli and Klebsiella, and they are so good at fermenting and capturing the nutrients from your food that once they've established, they're a wrecking ball. And we call them disruptors because as they grow, they disrupt the rest of the flora. Sort of like your garden is overtaken by weeds. And the weeds will win, the garden will lose. And that's what's happening. So E. coli is the biggest one. Klebsiella is less common, but worse to the microbiome. It's more disruptive. And then the others are minor. Overgrowth is not coliforms. It's the E. coli and Klebsiella that are there that now have the competitive advantage because of the stasis or different changes in the environment in the small intestine at that moment.
And this is some of the connection then too to IBS, post-infectious IBS and SIBO. These microbes are culprits.
Yeah, exactly. So we've shown this entire sequence. Like we've given food poisoning to rats, and then they develop the stasis of the gut. They develop these antibodies from the toxins of the food poisoning, the CDTB and antivinculin antibodies, because they react to the food poisoning. So you get this autoimmunity. And then the small bowel slows down from that. And when it slows down, the bacteria build up. And in rats, it's not Klebsiella. They don't have Klebsiella. It's E. coli. And E. coli goes through the roof. So we've seen this entire sequence in animals. So we know this happens. And we know that in humans, the only known cause and the only cause and effect cause, because this has been proven with the Bradford Hill criteria for IBS in humans, is food poisoning. Campylobacter particularly is the worst. And so it's been very well characterized. In fact, stress has been shown not to be the precipitant, but to be a cofactor. So stress, anxiety, and depression is not a cause of IBS, but is a modifier of IBS.
That's right. That's right. And many other things too.
And many other things. Blood pressure, heart disease, stroke, you name it, stress.
Inflammatory bowel disease.
Yes, exactly.
We got to all chill out.
That's right.
So you also had mentioned Lactobacillus as being a disruptor in a former conversation. And I'd love you to talk about that as your group's been really looking at the small bowel. Can you talk a little bit about Lactobacillus as a disruptor in the small intestine?
Yes. So, a lot of our work's been focused on irritable bowel syndrome and the relationship with overgrowth. And that's very exciting and it's really come to the forefront now. But we found some pearls in the REIMAGINE study that are super interesting. For example, is the Lactobacillus. So in patients who have unhealthy aging, so let me backstep. Lactobacillus has been studied, administering Lactobacillus, taking probiotics, has been studied on the basis of measuring stool. Nobody ever looked at the small bowel to see the effect of these probiotics on the small bowel. So now fast forward to the REIMAGINE study, we see that when patients have high lactobacillus in their small bowel, it is associated with more obesity, and it's very strong association. And then the second thing is we looked at aging and unhealthy aging and healthy aging. And unhealthy aging is associated with higher lactobacillus in the small bowel. Now, is it cause and effect? Those things need to be determined. So I can't say that, oh boy, because your lactobacillus is high, you're going to age badly. If you think about it, we give yogurt to our kids instead of fruit cups because we think it's more healthy. Is it right? Is it a good thing? We don't know the answer to it. But the third piece of that lactobacillus puzzle is we did a study looking at the disruptors of the small bowel. The more lactobacillus you have in the small bowel, the more the garden is destroyed, the normal flora. So that concerns me a lot because I don't know what the repercussions of that are, but you can imagine it might be similar to the E. coli and Klebsiella of IBS. The lactobacillus is not making you have all that IBS symptoms, but is it doing other things? Is it leading to obesity? Is it leading to other things that we need to understand more consequentially? And so more work needs to be done.
Yeah.
But I think that's worrisome to me. So I don't really advocate for lactobacillus or probiotics using lactobacillus in my practice.
Would you say that across the board for everyone? Like say you were talking to someone just interested in their gut health. Do you think lactobacillus has got a potential
I don't
problem?
know the answer. I mean, now that's really got me wondering what is good about lactobacillus? What could be bad about it? I don't know the answer to it, but it really raises a lot of questions and a few hairs on my neck because I'm worried that it could be harmful. I go back to what our mothers told us, too much of anything is a bad thing.
Yeah.
And that has always rung true for all of science, is that too much of one thing is always a bad thing. Oh, don't eat fat. Don't eat any fat. So, you swing the pendulum. So now we switch it all to carbs and everybody's getting obese. And now they're swinging back and saying carbs are okay or carbs are not okay. Fat is okay. And so the pendulum swings. But if you have a balanced diet, if you have a little bit of yogurt here and there as a healthy person, I think there's no problem with it.
Right.
But if you want to get 20 billion lactobacillus in your gut every day, is that healthy? I'm not sure.
Yeah. It's that whole notion that more is better. We just can't get away from that. Everyone thinks, well, geez, if 10's good, let's get 1 million and see what happens.
Yeah, if I can live longer because a little bit of selenium is good, more is better, right? So anyway.
It's interesting.
We have to remain curious. And I think that's the thing here is, and I think probiotics are a great example, right? That people are just, they want to feel better. And to your point, which one has the highest? If a little bit is good, then again, let me find the probiotic out there with the highest amount of that. And then they stay on it for too long without adequate relief of their symptoms. And it becomes a very muddy waters, or the garden is very heavily impacted by too many weeds and species.
Exactly.
But even if you're planting a garden, you don't plant all tomatoes. I mean, you can't expect one organism to correct 1,000, right? So it's like I say, if you add 1,000 lawyers every day to Los Angeles, that may not be a healthy thing for the city. We need lawyers, but maybe 1,000 every day is too many. So that's the concept of a probiotic. And not to mention the other sad thing about the word probiotic is it has the word or the prefix pro in it, like it's good. And then this notion, another myth of good and bad bacteria, That's a myth. There is no such thing as good and bad. There's bacteria are all good and all bad, depending on where they are. Lactobacillus in your bloodstream, not good. Lactobacillus a little bit in your gut, probably good. And so it just depends where they are and what circumstance. They all evolved to stay in their place and their niche. And when they are too high or in the wrong place, it's a bad thing.
Moderation in all things.
That's right.
That's it.
That's right. We've discussed, and you can correct me if I'm saying this incorrect. Is it Placide trial?
Mm-hmm. Yes.
Okay, so let's talk about the Placide trial. What did this trial look at, and how is this information clinically relevant for us?
So this was a study looking at patients that were given antibiotics for whatever, a urinary tract infection or some sort of infection at the moment in primary care clinics. And then as they were given the antibiotics, they were randomized to probiotic or no probiotic. And what they found was there were no decreased rates of C. difficile or antibiotic-associated diarrhea because you're on the probiotic. That was what they were hoping to see, is that would prevent C. diff or something like that. And the only thing they found that was statistically different, the only significant thing really, was that the probiotic group had more bloating. So this is what I tell my patients: if you want more bloating, please take a probiotic because it will provide that for you, because the Placide trial is the largest randomized controlled trial to date on this. So it kind of settles the issue. Now, the probiotic world is a plethora of products. So it's very easy for the company to say, well, that's not our strain. Our strain doesn't do that. Or that's not bifido, that's lactobacillus. Or, so whether I say this or not in a podcast, and whether somebody hears this or not in a podcast, the product companies are going to say, well, that wasn't my product. And so the story continues as we unfold this. But the problem is that these companies don't have the money or don't want to do these incredibly large trials to prove one way or the other if their product does what they hope it does. So it's a tricky area. I do believe bacteria are important. I do believe there are some that are better than others. I just don't think you got to jack up the system with too much of it.
Especially if the small bowel is not working very well. It just feels
Even
like you're
worse,
just
right.
accumulating.
I can tell you, absolutely, I have patients who have IBS, who've taken probiotics and said, oh my God, I feel so much better. It's rare, but it happens. And so I say, great, call me when it's not. Inevitably, they call me and it's not working again. And then we go back to square one and we go through the fundamentals again. But, I can't deny that there are occasions where patients feel better, but anecdote is not science. We deal with it in the clinic all the time.
Yeah, it's true. Especially when you really want to feel better. You're hoping so hard on that little probiotic capsule.
Exactly.
So let's talk a little bit about SIBO and new treatments. And I know your group reported at DDW this year about using rifaximin with N-acetylcysteine or NAC, and found that this combo worked better than rifaximin alone. I know rifaximin has FDA approval for IBS-D, so kind of used off-label for bacterial overgrowth, but is used commonly. So what led you to looking at NAC and rifaximin together? Like, what prompted that idea?
So when we helped develop rifaximin in the first place, it is a great drug for IBS. It's the number one drug for IBS worldwide now. If you go to ChatGPT, you can ask, because it's safe, because 2 weeks might get you 6 months of benefit, which no other drug does. So in that context, it's been very successful. But I was never satisfied, because what I'm seeing in the microbiome of IBS, more people should get better from rifaximin. Something didn't add up to me, because we're seeing more SIBO in IBS than rifaximin makes better. So, for example, for rifaximin Target 3 trial, 44% of people responded to rifaximin, but we're seeing 60 to 70% having SIBO. So why are the others not getting better, or why isn't it as good as it should be? Where rifaximin's characteristic are, it's not absorbed because it's hydrophobic. It's like an oil almost, technically. It doesn't have an ability to dissolve in water. So there's that part. And then what we found in the REIMAGINE study in SIBO patients is that E. coli and Klebsiella, those two bad disrupting characters, those Ferraris, when we calculate where they live in the small bowel, 50% of them live in the free fluid, which rifaximin has access to, and the other 50% live in the mucus, not the mucosally associated, but the mucus, the thick, stringy stuff. And rifaximin will never get into that. So we did some studies preliminarily to see if we added a mucus buster or, a mucolytic like NAC, does rifaximin work better? And it works 10 times better in that situation. So we are able to get rid of the Ferraris more comprehensively if NAC is present, and we can use a much lower dose of rifaximin to get the same effect. So we did an initial study, which we presented at DDW, and it showed that in a very small study, rifaximin and NAC is better than rifaximin, the one on the market. And so the phase 2B study, I just had a meeting before this one, we're kicking that off in September. It's the first phase 2B study at our center that isn't pharma funded. We're doing the whole thing ourselves to see if we can get this better for patients. So it's pretty cool. No pharmaceutical company.
No pharmaceutical company. Yep. Amazing.
Why is that important for a listener? What does that mean?
Doctors do a lot of things with pharmaceutical companies. We try to get things across the finish line, but I'm not doing this for that. I'm doing this for patients. We want patients to get better. This isn't about pharmaceutical companies. This is about doing the right thing and finding the right answers and getting these things for patients. So I'm hopeful this will amount to something and then make patients better more often. I believe it will. Our data has shown that. We even used our rat study, the rats who were post-infectious, and sure enough, just giving rifaximin versus rifaximin and NAC was completely normalized their cytokines, normalized their microbiome, normalized their bowel movements, whereas the rifaximin only partially did that. So we're going to the next level, and we're doing it alone.
I love that. So just quickly, how much NAC? Are you using the same amount of NAC that you used in the trial that was reported at DDW? Can you disclose the amount that you're using in the trial?
So it's a different formulation than that trial. Now this is the final formulation where it's delivered in the small intestine, specifically in the locations where the E. coli would be expected to be. So it's got a special delivery system. It's different than the other.
Okay.
So we'll see how it goes.
Awesome.
We'll be waiting.
Yes, we will.
And I will have no fingernails left by the end of it as I'm biting
I bet you won't.
We appreciate all the work you're doing, especially in this area, because you're really it seems like the one and only that's just really knee deep in it and pushing the bar.
Abdominal pain and diarrhea from IBS-D getting in the way? You may find relief with Xifaxan with an X. Xifaxan rifaximin is a treatment for adults with irritable bowel syndrome with diarrhea. Visit Xifaxan.com/IBSD for the PI or talk to your doctor. Don't use Xifaxan if you have a history of sensitivity to rifaximin, rifamycin antibiotic agents, or any components of Xifaxan. Tell your doctor right away if your diarrhea worsens while taking Xifaxan, as this may be a sign of a serious or even fatal condition. Tell your doctor if you are pregnant, plan on becoming pregnant, are nursing, if you have liver disease, taking warfarin or other medications. Some medications may increase the amount of Xifaxan in your body. Most common side effects are nausea and an increase in liver enzymes. Xifaxan.
So let's switch gears to methane. And I know there's been a number of different studies linking high levels of methane with constipation. But I'm wondering, is this bidirectional at all? If you're constipated, does that contribute to methane being produced or more substrate for those microbes? Is there any connection with just say someone's just constipated, doesn't have elevated methanogens? Do they develop elevated methanogens just because they're constipated? Or is it just the methanogens are causing the constipation through their methane? Does that make sense?
Yes. And that's an argument that's resurfaced recently. And I wonder whether some of those folks hadn't read the earlier papers that have already been done, because we've gone through that argument already.
Okay.
So yes, if you have methane, you're more likely to have constipation. The higher the methane you have on the breath test, the more constipated you are. It's very proportional, which is really important for cause and effect. But that doesn't mean that the methane is causing the constipation, because if you're more constipated, maybe you have more methane. Maybe the methanogens love that dry stool environment and they do better in that situation.
Yeah.
But we've done the other side of it, where we've given methanogens to animals and they get constipated. So we gavage them with the methanogens. We've done the studies of live animals where we infuse methane into the small intestine and they get 60% slowing of intestinal transit. We've done organ baths of the smooth muscle of guinea pig ileum, adding methane to the bath. And the guinea pig ileum, the peristalsis goes away. It goes into more of a spastic contractile activity. So it's the methane. And we're now all of a sudden, maybe there's a study that says that, I don't know if that maybe they're constipated first, then they get methane. I'm not sure. It hasn't come out yet, but this is coming back to life. But maybe they should read those papers, because I think we pretty much nailed that down, that methane is a constipating agent, period.
Yeah, I thought so too, but I was on, as I do, lurk on Twitter and saw a recent paper, and I'm like, I'm going to ask Dr. Pimentel about that.
Well, the paper hasn't come out yet, so it remains to be determined.
Yeah, I just saw the commentary.
But I certainly hope that they at least reference those papers, because those are the ones that show that it's primarily methane that's causing the constipation and slowing the gut down. But it's okay.
You heard it here first.
And in more cutting edge research that's coming out of your lab, let's talk a little bit about CS06. So you presented a preclinical study looking at the molecule in relation to reducing the methane gas levels. And this is, as we've really identified, methane associated with constipation and also EMO in human studies. So what is CS06 and its potential mode of action? And based on the timing of the research, if this study proves itself to work in humans, when do you think it's going to come to market?
Well, this is more proof for the last question. So we did a double-blind study of rifaximin with neomycin with methane, and only the humans, or most prominently the humans where the methane went down, did their constipation resolve for a period of time as the methane was gone. So it wasn't the effect of the drug, it was the effect of the removing the methane. Again, proving that methane is the issue. So now we have CS06, which blocks an enzyme in the production of methane by the Methanobrevibacter smithii bug, or archaea, that's causing the methane in humans. And we see 70% reduction in methane with this drug. And the study we presented at DDW was the controlled trial in animals, where these are animals that we can make constipated, make their methane go up, that we spoke about in the last question. And then we give the drug and the methane goes down and the constipation goes away. So again, cause and effect of methane equals constipation. So the animals had less constipation and less methane. So we are about a year away. There's all, you got to do all these toxicity studies, et cetera, to get to humans. And so we're hopeful to be in humans within a year. And that will be very exciting, because then we're really treating the cause, right?
Right.
It's not an antibiotic. It's not a sledgehammer. It's just a small molecule to make the bug stop doing what hurts you. And that's it. So that's very exciting.
Very exciting. So back to methane. As a dietitian, we're always working with patients with EMO and want to help them. And there's been some talk that a low-fat diet would help with methane production. Is there any diet interventions that you're finding useful in your patients, or that you have data to talk about?
Well, we do know that high-fat diet does promote methane production. We see that in animals too. If we gavage them with methanogens and give them high fat, the methane goes up a lot more than if we didn't use high fat. Don't know why, except we know that methanogens use bile acids as a fuel source as well. So more fat, more bile acids. It's possible that those are the connections, but we do see that happening. We don't necessarily see it on high protein. High protein can cause more constipation as well. We don't necessarily see more methane in a high protein diet.
Interesting.
But we haven't gotten around to saying, please go on a low fat diet. This will make your methane go away. We haven't done that.
Yeah.
It's an interesting exercise. I wonder whether it might help, and if we get to hydrogen sulfide, there may be things that can be done there. I almost see that because what we've identified in IBS, if I can go back a step, is 3 microtypes, really. There's the SIBO microtype with this E. coli, Klebsiella, 2 Ferraris racing to get all your food. Then there's the methanogens, which are the constipation side of things, and their needs are different, and maybe there's a diet for that.
Yeah.
And then, of course, hydrogen sulfide, when it's there, you get more florid diarrhea, and they need sulfur. So if you had a low sulfur diet, maybe that would be helpful. So I think diet's going to play a role in all 3 over time as we understand their nutritional needs and what's driving their increased metabolism and growth. And so diet may play a big role in all 3.
So who's going to do those studies?
Well, I can't do all the studies, Kate.
I know. Maybe Bill Shea.
Maybe Bill Shea. Let's get Bill.
Bill.
You know, it's like, what do they say? Let Mikey eat it.
All right. We got to find someone to do those studies.
And it might happen. We've got Dr. Prashant Singh. We've got Dr. Che here at the University of Michigan. So I don't know. Here's our call outs. I'll go knocking on some doors.
Bill's an expert in diet studies. So
absolutely.
Anyways, I've talked to him and he's got a lot on his plate, but I wasn't trying to commit him to something. But
I'll commit him. No, we'll get Prashant. We go for the younger ones, that are coming up.
All right. So you mentioned the 3 gases, right? We've got the hydrogen, the methane, the hydrogen sulfide, and we now have testing to look at all 3 of these gases via breath tests, and they're available nationwide. And you did a real world study, with a rather large cohort of people, over 3,000 that completed all aspects of the study with questionnaire feedback. So tell us a little bit about what you learned, because again, these things that are available on the market, people are interested in, and we want to know what they show us and how it leads to treatment.
Well, we do it for the sake of science, trying to understand these different subcategories of gas types, but we also do it for critics because always there's critics. People said, breath testing is great, but now you got to do culture. Then you do culture to say IBS has SIBO. You publish that. They say, yeah, but next generation sequencing is better. We don't believe it until you do next generation sequencing. Then we do sequencing and we show the same thing. And they say, yeah, but you could do shotgun sequencing. And so now we've done shotgun sequencing. We published that. Anyways, the list of the critics' desires never ends. But we keep showing the same thing with the different tests, and the same thing with breath testing. So, we show that it works in clinic. So methane is associated with constipation. Hydrogen is associated with diarrhea. In-person breath testing at Cedars-Sinai, for example, or at Ann Arbor. But now you're doing mailing at-home breath testing. Does that work? Can patients actually do it at home? And a 6,000 breath test study says, absolutely. Methane's associated with constipation, SIBO and hydrogen sulfide is associated with diarrhea. It all turned out the same way, and the results were equally impressive. But what we were able to show is that these gases interact because this is such a large study. There are some patients who have all 3, and that's pretty remarkable. But what I used to think is that methane is the winner. Like methane will because of its constipating effect, it will out-constipate the diarrhea, but not with hydrogen sulfide. Hydrogen sulfide is the winner. Anytime it's present, it is dominating the symptoms and making all the symptoms more severe, and in particular, visceral hyperalgesia or pain. So hydrogen sulfide is an activator of pain pathways. Then what we also did at DDW, which I don't think we were going to get into, is we actually looked at the biopsies in the REIMAGINE study of the bowel, and the same thing in the animals where we gavage these organisms to test whether hydrogen sulfide is causing diarrhea or not. And of course it does. The changes in your lining of your intestine when you have these hydrogen sulfide organisms are incredible. It's literally, you're killing the cells almost at certain levels, but you're causing visceral hyperalgesia, intestinal barrier proteins are affected, serotonin is affected, mitochondrial function is affected, so your energy of the cells are affected. H2S is nasty. It's nasty. But anyways, in the 6,000 patient study, we were able to confirm that hydrogen sulfide, the higher it is, the more diarrhea, the SIBO and the methane in this very large scale study.
So interesting. And wasn't like hydrogen sulfide connected with inflammatory bowel disease? Didn't they think that like years ago? I haven't really heard a lot recently,
Well,
but
what happened with that? So there was this big movement in the early 90s of the relationship between hydrogen sulfide in the colon
yeah.
and the development of ulcerative colitis, for example. It was ramping up, so to speak. And then infliximab came and boom, everything changed to biologics. And it, in essence, cut off at the pass the continued work on hydrogen sulfide. But maybe now it's going to be reinvigorated in IBD too. I know there's a few studies that are ongoing already looking at ulcerative colitis and other IBD patients with H2S. H2S is toxic, so it's not good.
Interesting. And really, there's only one test. Like most hospital settings are not testing for hydrogen sulfide. It's this mail order.
So it depends on the hospital system. There are some. Stanford does. I
Okay.
think Bill Che is going to start. I'm not sure. Northwestern does all three. So
Okay.
it just depends because you just get the kit. You can do it at the academic center and send it. So you can do them in person still. You just have to have the kit.
Yeah.
Or you can do it by mail order.
That's amazing. You know, I've been doing this for a long time. And in the beginning, it was like this big thing, request hydrogen and methane. Like that was a big deal because they weren't looking even at methane, you know, back in the day. And now it's like,
well,
okay.
I think the biggest problem we had, even in SIBO's been around, breath testing's been around since the 80s. And even centers where they're somewhat critical of breath testing, they're doing tons of breath tests because it makes money for their institution. To be honest, that's sad that you would be a critic, yet you're still doing these tests. Anyways, the tests do work well in my view. But people started pulling these Quintron instruments out of their basement and turning them on and starting to run them. They need servicing. I mean, these instruments, these gas chromatographs need servicing. Yeah. And we've experienced some pretty high level institutions where the breath test isn't calibrated properly and they're getting wonky results. So it's a delicate test to do right and therefore get good data. So that's another problem that we've encountered over the years.
Yeah. So listeners, ask about what equipment they're using and maybe inquire about the 3 gas breath test. So as we wrap up, I wanted to just briefly talk about fiber. I know a lot of the work you do is in IBS and SIBO. Is fiber a friend or foe, or is the answer kind of somewhere in the middle?
Like everything else, your mom told you, keep everything in moderation. There was a study that was done in Japan where they fed rats kidney beans for 2 weeks. So that's high fiber.
That is.
The rats developed SIBO just from eating kidney beans.
Yeah.
So beans, if you were to be on an all bean diet as a human, you're going to get SIBO, even if you don't have any underlying reasons for SIBO. That is just an example of what high fiber can do and the power of high fiber in terms of SIBO without any other medical problem. So too high fiber is not a good idea. Too much of anything is not a good idea, right? That's the theme today. But with people who already have SIBO, you're basically putting logs of wood on the fire in a sense, because now you're providing more nutrients for the Ferraris to be able to produce more gas, bloating, and distension. So the offset of that is, yes, fiber can make you have more bowel movements. So maybe if you have more bowel movements, you clear some of these bacteria out. Maybe that makes you feel better because you're having diarrhea from the fiber or whatever the manifestation is of the fiber. But all in all, we see that fiber just causes a lot more bloating for these patients. And so we generally recommend low fiber, not no fiber, just low fiber.
Yeah. Incidentally, the kidney beans are very, 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 like probably the highest fructan containing bean. So it's a very high FODMAP, small chain carbohydrate, you know, even compared to like chickpeas or lentils or black beans or cannellini beans. Kidney beans are like the top dog. They're so concentrated. So we're giving them a lot of fiber. And I do wonder, like in bacterial overgrowth, if the smaller chain fibers are more problematic because they're easier, they're rapidly fermentable. And, you know, are they going to be in that small bowel a limited amount of time? So those things they can access a little quicker are more problematic. Just, you know, critically thinking. I don't think, in my opinion, I'm not sure that all fibers act the same or maybe as problematic for SIBO as maybe some that are really quickly accessible by our microbes in the small bowel. But tell me otherwise.
So I like to do everything based on science, right?
Me too.
And I don't know that there's enough science to tell me it's got to be this long, that long, this long, that long. All I know is it's fiber or no fiber. And that's really inappropriate for me to be so dogmatic, but I don't have any data. All I know is if the patient is on fiber, or if they're on Metamucil, or if they're on some product that is fiber containing, which is good for some people, it isn't good for SIBO. It just, the patients do poorly. But I would love to see a study where you compare different fibers to see which ones they tolerated. But again, that's not been done.
Prashant Singh calling.
We're volunteering him for a lot of studies.
We are.
Yes, we are. Yes, we are. And clearly, I love the debates. I love the conversations. It really is important. As I said earlier, we have to be curious about this. We cannot just lean into, it's not black and white is the reality. It's not black and white.
It's never black and white. And that's why, people who are overexpressing, I like to tell you things that I have a ton of data on. Mm-hmm. And I warn you of things that I'm cautious about, but I clarify. I don't have data. I can't tell you what fiber could be good for SIBO. I just don't have data on it.
Right.
But so my overall gestalt is low fiber for now until we figure it out.
All right. Well, you told us last year that in order to navigate, the stress of running this massive lab, you are a blues guitar player.
Oh my God. Did I tell you that?
You did tell us that. And I've been waiting for, an MP3 or something of hearing you play. But what are you doing for your well-being and self-care these days?
Well, I have a home up in Banff area, so I go up there and I write papers up there and just spend a little bit of time with nature. It's very good. It's very cleansing. I also have guitars up there.
Perfect.
That is a magical, magical place. We were in Banff a couple of years ago. It is one of the most beautiful places I've ever been. How lucky.
Yeah. Now, we've gotten to know it quite well. And if you know it quite well, you know the real places. Yeah. Because Lake Louise is beautiful, all of that. Yeah. But there are places where hikes could be 5 miles up to a lake where there is literally not a human anywhere. These mountain lakes, and they're just spectacular. The water is like glass, and there's nobody.
Love that. We went when it was freezing. So to be honest, there was really no one there except for us in my poorly dressed attire because I was so cold. But it was kind of nice because we missed the crowds, but, it was a little chilly.
The crowds are too much, but that's okay. Same everywhere.
Exactly.
That's the key. You got to get away in order to kind of clear the brain and just give yourself the space. And so a little slice of heaven, it sounds like. That's incredible.
Yes, you deserve it. So thank you so much for joining us. We really appreciate your time. And the first podcast that you joined us with is still our top producing podcast. It's got the most downloads. So no surprise. People are really interested to hear your brilliance and expertise, and just you're really making a difference in so many people's lives. So thank you. And thanks for your time today.
No, it's my pleasure. It's good to talk to you both.
All right, friends. Well, don't forget to like, share, and subscribe to the Gut Health Podcast. Your support means the world, our friends. Thank you for joining us as we grow this gut health community. We hope you enjoyed this episode, and don't forget to subscribe, rate, and leave us a comment. You can also follow us on social media at the Gut Health Podcast, where we'd love for you to share your thoughts, questions, and experiences. Thanks for tuning in, friends. Thank you for joining us as we grow this gut health community. We hope you enjoyed this episode, and don't forget to subscribe, rate, and leave us a comment. You can follow us on social media at the Gut Health Podcast, where we'd love for you to share your thoughts, questions, and experiences. Thanks for tuning in, friends.